When a problem arises, people turn to scientists for answers. The University of Washington 2006 Science Forum features faculty sharing research results on the world's most vexing problems. Mary Desis examines how the immune system may be used in the future to prevent cancer through the development of vaccines. David Eaton looks at the research on genetic, lifestyle, dietary, and environmental factors to puzzle out why some of us get cancer and others don't. Garrett Odell examines the use of mathematical models to shed light on how biological gene networks operate and how these models reveal the networks to be astonishingly robust, despite their unintelligent design. Buddy Ratner updates us on the progress of regenerative medicine and addresses the practical and ethical issues surrounding tissue regrowth and organ replacement. And Ray Hilborn provides facts to dispel the fears of a worldwide fisheries crisis and discusses the changing objectives of fisheries management. Join us in Puzzling Out the Science. Exploring Our World, Puzzling Out the Science is made possible by the University of Washington Alumni Association, the College of Arts and Sciences, the Office of Research, the College of Engineering, the School of Medicine, the College of Ocean and Fisheries Sciences, and the School of Public Health and Community Medicine. Additional funding for this program is provided by the University Bookstore and UW-Medicine.
Welcome to the 2006 Science Forum. I'm Ron Irving. I'm the Divisional Dean of Natural Sciences in the College of Arts and Sciences. I've been involved in this series for several years now. It's a wonderful series. It's a great opportunity for us to hear about some of the best faculty at the university and their exciting research. And it's a great chance to learn about how the universe works and our place in that universe. Tonight's speaker is Gary Odell. As it says there, he's a professor of biology here at the University of Washington. He's the director of the Center for Cell Dynamics, which is up in Friday Harbor at the University of Washington's Friday Harbor Labs. The Center for Cell Dynamics fosters research in cell and developmental biology that fuses experimental approaches with computational methods to better understand complex biological systems. We'll be hearing more about that in the lecture tonight. Professor Odell began his career as a mathematician, which is kind of exciting for me because I'm a mathematician. And as I said to an audience earlier, I get to see from him what I should have been doing perhaps instead of what I do. He got his bachelor's and PhD at Johns Hopkins University, studying theoretical mechanics, and then spent 13 years in a math department at the Rensselaer Polytechnic Institute in New York. He has not always been a biologist, in other words. He's basically a computational modeler. And again, we'll see more of that tonight. And what he wanted to model, biological systems, we weren't able to do years ago because we didn't have fast enough, good enough computing machines. And with the advent of more advanced computing, he was able to realize his vision, and let me quote from one of his websites, his vision of applying mathematical and computer modeling techniques to produce, from true molecular facts, real explanations of how molecules animate cells, and thence how leaderless swarms of autonomous cells self-organize to form embryos that grow up to squirt the genes responsible for their behavior into the future. It's a stunning statement. So from math to molecules, to cells, to genes, to our future. Professor Odell has carried out this vision since 1986 here at the University of Washington, first in the Department of Zoology, now the Department of Biology, which zoology merged into, and again at the Center for Cell Dynamics. He'll tell us more about his vision in his lecture tonight titled, "For Making Genetic Networks Operate Robustly, Unintelligent, Non-Design Suffices." Please welcome Gary Odell.
Well, thank you very much to the University of Washington for inviting me to tell this tale. Look at this miracle on the screen. A giant hawk moth laid this egg, provisioned it with a great deal of nutritious yolk, and put one nucleus in it. And that nucleus divided over and over and over again to make thousands of nuclei. Then messenger RNAs and proteins synthesized from the genes in those nuclei self-assembled with those very genes into a number of genetic networks, which working in concert convert that yolk into this worm that hatches after 100 hours with guts and legs and a brain and an attitude and an insatiable hunger. The first thing it does is eat its own eggshell, and then it eats everything in sight. And within 14 days, it grows to the size of one of those Schulze's sausages available over on the Ave and metamorphoses into a hawk moth that lays eggs. Now, all of this is brought about by the action of genetic networks, and that's what I want to talk about tonight. I want to talk about new work in which we probe how these gene networks have acquired a phenomenal robustness they have, an ability to continue to operate correctly in the face of all kinds of perturbations. That's new and unpublished work, but to set the stage for it, I have to tell you about published work involving the revelation that gene networks could be as robust as I would like to explain in the second part of the lecture. To ground that work, I have to tell you first about what gene networks are, and to do that, I have to tell you about the central dogma of biology, which I hope is a review for everyone. For this material I'm about to go through is the stuff from which a creation myth will spring, which is as sublimely beautiful as the ancient ones, with the added benefit that it's probably literally true. Now, this is a cartoon of a cell, and to produce that larva from its initial first cell, it becomes 50,000 cells that differentiate one from another. In other words, even though they have the same nucleus here, they acquire different states of gene expression that makes them different one from another. They do that in a spatially patterned way, and the buzzword in all literature for understanding this phenomenon is to understand pattern formation. So I'm going to tell you the molecular foundations of this pattern formation phenomenon. So here is one cell, and each cell has its nucleus, and in this nucleus are chromosomes, which are very long strands of DNA, single molecules, along which genes are arrayed. A gene is some stretch from this red dot to that red dot, for example, and there are thousands, or for humans, tens of thousands of these genes in each nucleus. Here are some nuclei in a fruit fly dividing synchronously, getting ready to make the caterpillar that hatches from that egg. Now, under certain circumstances, machinery made from proteins, called DNA polymerase, attaches to the DNA and starts copying this DNA into RNA, a different kind of nucleic acid. So this purple strand is a copy of the gene. And then the next thing that happens is that messenger RNA is exported from the nucleus, usually after some splicing to get rid of introns. And that RNA is then translated by ribosomes. Those are particles made from the proteins I'm about to tell you about, along with other pieces of RNA. They translate this RNA into a protein sequence of many amino acids, and those amino acids fold up into a three-dimensional high-tech nano device. Now, you should think of this device as something like this Leatherman, okay? It's got grippers, and it's got little blades and all kinds of things sticking out from it, and it is a potent device that can do all kinds of things. There are thousands of different kinds of these devices, and of each kind, thousands or up to millions of copies in a cell. So to imagine what's going on in a cell, you might imagine all of those confiscated penknives in all of those boxes at airport security checkpoints, all gathered from all over the world and poured into this room to fill it to the brim. And imagine all of those millions of confiscated knives shrunk to the size of a few nanometers, vibrating around, bumping into each other, and interacting. So these proteins are capable of an astonishing range of capabilities. Here I've cartooned that some of these proteins re-enter into the nucleus, attach to the DNA, and actually turn on the transcription that I started the story with. So this, if this is what was going on, this is a gene whose protein reinforces its own synthesis. It's an autocatalytic gene, and I've drawn how it ramps up the rate of making the RNAs. Now, these proteins can do all kinds of other things. They can be single motors that run along tracks in cells. They can be the building blocks of the actin and myosin that work your muscles. As I said, thousands and thousands of different kinds of things. Here I've cartooned one that makes a protein inserts into the membrane of a cell to be recognized by some other protein in the other cell so that this cell can send a signal to that cell. And in addition, these molecules can sometimes hopscotch onto the membranes of adjacent cells. Now, this is important for what I want to say later, that most organisms, such as us, such as that hawk moth larva or Drosophila, are diploid. They have in each nucleus two copies of each chromosome, and that means they have two different versions of each gene. They have an allele from their mother and another allele from their father. Allele is the word you need to name alternate sequences of the same gene. And what happens over time is that these sequences diverge by mutations, so that in a population there may be tens or hundreds or thousands of different versions of each gene, different alleles. And when an offspring is produced, those alleles are at random selected from the mother and father's genome to produce the offspring's genome. A population with lots of different versions of each gene is called polymorphic, and I will talk about that some coming up. Okay, so few genes do anything at all by themselves. There's a few, like superoxide dismutase, that makes a free radical scavenger, but very few genes do anything by themselves. Instead, networks of genes conspire to perform each essential task, and the different alleles of those genes means that the different groupings into networks of genes are different quantitatively, one individual to another. And I think that in natural selection races, it's the ability of an allele to collaborate with the other genes it's involved with in networks that boosts its chances or hurts its chances for prospering in the gene pool. Now, here I'm showing in this time-lapse movie of a live Drosophila embryo, green jellyfish fluorescent protein driven by the promoter of one of these genes listed down here, the engrail gene, lighting up stripes that form in this larva as it develops, prefiguring the body plan of the larva. This will be a segmented larva, and these stripes here lay out the coordinates or the pattern of segments that will form morphologically in a bit in this embryo. This network I'm first going to talk about, it's called the segment polarity network, and its genes are named engrailed, wingless, patched, hedgehog, cubitus interruptus, frizzled, disheveled, beta-catenin, and pangolin, and there's a few more. And these genes, by the way, are operating in you as well as in these various arthropods. So I'm now going to tell you about the icons I'm using in my cartoons of genetic networks, not so that you'll memorize them, but so you'll get an idea roughly what a genetic network does. This is a picture of one. And in it, these round icons represent messenger RNA, the nucleic acid from which the proteins are translated, as in this step. These hollow icons, the octagons, represent proteins in the cytoplasm soluble. When they have a bright fill, it means they're stuck on the membrane of cells and can diffuse cell to cell. When they have a dim fill, they're stuck on the membranes. These arrows leading to an RNA indicate turning on transcription of the gene, and these dots indicate repression of it. This, for example, means this transcription is always on, and all these arrows and lines say how each gene is connected and each gene product is connected to the others. Another few icons, sometimes two proteins like this one and this one will stick together to make a dimer, a composite protein, and these little crow's feet indicate that. Some proteins, always with a diamond-shaped icon, they act to convert another protein into still a third protein. So these are things like enzymes and kinases in my diagram. So that's what these diagrams show. Now, I want to say that we are all made and operated by genetic networks, and they're assembled from the same pool of genes common to a huge diversity of organisms on Earth. That's a big surprise from the last 20 years in molecular biology, so that we can study what these networks do in fruit flies and in organisms that that develop rapidly and are convenient to study, and infer what's going to happen in humans. Now, the point of this slide is to say that every network must be robust. And what I mean by that is that when the network happens to be randomly put together from alleles, different alleles from randomly chosen parents, the network still has to work in spite of the differences between the alleles. So this is a simple calculation to just say the probability of a typical network failing as a consequence of any perturbation it's likely to see in your life is very low. If this is the probability of it failing, and that's about the number of networks operating in a human, the probability of it failing needs to be less than 1 in 1,000 in order that you're healthy 70% of the time, or 70% of the people are healthy. It's rare that one of these networks is going to fail. They have an amazing robustness. Now, these slides give you a hint about how to think about genetic networks. Here's a really boring one in which the input A, when on, turns on the output E, and when the input's off, it turns off the output. It's like a complicated wall switch with a lot of gears. So it's easy to figure out, and it's boring because the output is the same complexity as the input. How it works is that if A has been transcribed into RNA, that protein made from it feeds back to reinforce expression of A. When A is on, it turns off B. When B is on, it would suppress C, but when B is off, it allows C to be made, and C is then converted to E by D, which was also turned on by A. So you can figure this out just looking at it without any mathematical modeling. But as soon as you add this kind of feedback that I've just put in here, so that E suppresses A, then it becomes an imponderable what this network is likely to do. This network encodes the Russell paradox, namely A is on implies A is off. If you trace through the logic, as before, if A is on, it creates E, and that turns A off. So it's very difficult to figure out what such a network might do without making a mathematical model of it. And here is a picture of the segment polarity network coming up. Here's the one I was just drawing. These networks are capable of making truly remarkably complex patterns and doing it robustly. So the idea is that if you were to install a copy of this network in every one of these hexagonal cells, all identical, and all talking to each other across their membranes by virtue of, in this case, this diffusing protein A, then this network over time could bring about a sequence of different cell states. The different colors represent different concentrations of protein in the different cells, and I'm going to use that throughout this talk. Different colors mean different expression levels of one or another of the genes in these networks. So this network could produce a sequence of state changes of the cells and eventually, let's say, stabilize this pattern. This network happens to do that. And the question now is how do the cells know what to do? How do they put together this coherent temporal sequence? So it's Isaac Newton pondering how do the planets know what to do next, who provided, I think, the most elegant answer for that planetary question, and in the process answered all similar questions, at least in principle. So the idea is that any state, like a number of cells expressing a number of proteins, is characterized by the state of that system, which in the previous case might be the concentrations of all the proteins in all the cells at a given instant. And what Newton's idea is that the time rate of change of that state must be a function of exactly the state of the system at that instant. It's a tautology in the sense that what else could the rate of the change of the state of the system possibly depend upon other than its current state? So in any case, this word equation is written down mathematically, a differential equation. And if you dare to specify the state of the system, like Newton did for the positions of the planets, and specify the rules by which that configuration exerts the forces that changes the current state of the system, you have an equation that you can solve, at least these days you can solve it with a computer, to forecast what unfolds in the future from the current state of the system. And it's that kind of modeling that we do to understand the dynamics of these gene networks. So here is a 3D reconstruction of the stripes of engrailed, that's one of the segment polarity genes, in a fruit fly embryo. And this, analyzing the network that is known to make these stripes is the topic of this paper published about 4 or 5 years ago, which sprung from the dissertation of George von Dasso, which he did in the biology department here. And Elie Meyer did a further study and also helped with this one I'll first talk about. And Ed Monroe did a great deal of work on these two studies coming up. So we tried to figure out how it is that the well-known network of genes actually does make these stripes and what the property of those networks were. I should mention that this network operates in many diverse arthropods cued by different genes upstream than is the case in Drosophila. Okay, so here's the network, and you know, unless you're an aficionado of these kind of things, you should ignore the details, but just get a sense that it has, you know, 10 or 15 different gene products that interact with each other. This is really, this diagram here is not a fiction. It took something like 3,000 person years to do the experiments to reveal this network and certify every one of its connections. So we're really certain of every one of these connections, although some of them have been simplified in this diagram. It involves genes that, by the way, operate in you, hedgehog and wingless, which diffuse from one cell to another. It involves a gene patched. It involves all these genes that I've named. Now, while we know all these connections for certain, we do not know any of the rate constants. We don't know how long one of these proteins survives before being degraded. We don't know how much cubitus interruptus protein it takes to turn on what rate of synthesis of patched RNA. We don't know any of these quantitative details. Nobody has measured them. We presume that there's some curve that tells how, as you increase this protein, the rate of synthesis of patched goes up. But we don't know what the curve looks like. What we know for sure is it doesn't look like this. It's not the case that the more cubitus interruptus you have, the faster you make patched without limit, because these are biological interactions. They must saturate. In other words, you can't make the patched protein faster, patched RNA faster than some limit set by the transcription machinery. So we presume there is some curve shaped something like this, but we don't know what it looks like. We introduce parameters, 3 of them here, to sculpt what that curve looks like, the slope here, the point at which this curve reaches half maximal saturation, and what is that maximal saturation. And we think of those parameters as dials that we could turn, and by turning them, we could convert the green curve into the purple curve, into the red curve. We could draw any curve at all that turns out to be the real one by turning these parameter dials. It takes about 50 of these parameters, 48 of these parameters, to quantify all the relevant interactions and half-lives in this network, of which nobody knows any values. I want to introduce a metaphor. I want you to think about the first transistor radio ever patented. I actually owned one of those. That's how old I am. And this radio, whose idea is in the circuit, is in how you hook these capacitors and resistors together topologically to make something capable of resonating with KUOW. The idea resides in the wiring diagram, just as the idea of how this network works resides in this diagram graphed here. The patenter, what's his name, Koch, thought he had better write down all the resistances and all the parameter values of the capacitors and so on, because in fact, without those values, it's not easy to make this radio work. It may not even be possible to make this radio work. So how big is the parameter space? It's huge. If you think of a 50-dimensional parameter space, and imagine just trying out empirically at random high, medium, low values for each of the parameters independently, if you figure out what 3 times 3 times 3, 50 times in a row is, it's 1.2 moles. It's this many different, quantitatively different versions of that network that you would have to try just to do this very crude exploration of how it might work as a consequence of different strength connections between the parts. Now, this is, of course, not very promising for radio design. I can tell you from experience that if you tinker around and put in the wrong resistor, you get smoke and you know, you get PCB smoke. the radio not only doesn't work, it burns. It is also daunting for natural selection. How is natural selection going to try out this many versions of some network? There are very few species of which a mole of individuals is ever made. There's a few species, but very few. So this was our into this problem. What we did, in the absence of knowledge of what the parameter values are, we decided we would compute what they must be in order to make the network work. We failed to do that, as I'll say, but what we tried to do is install in every one of these hexagonal cells an identical version of this network. We used periodic boundary conditions. We then picked random values for the parameters, the 48 parameters in it, and then we solved the hundreds of differential equations that keep track of how the concentrations of each protein, each gene product, change with time in each of these cells. And we could ask whether, given a start nearby this end state, this network would stabilize the pattern in which engrailed is on in this green row and wingless is on in this red row, and so on. Whether the network could make the pattern that the fruit fly does make, is known to make with this network. We wrote a computer program which is called Ingenue. It's available to download here. And what that does is let you describe the network on a piece of paper, in a text file, and this program instantiates it into every one of the cells. And then it lets you set the initial conditions, the conditions at the beginning of time here, and it computes the solutions to these differential equations and shows how all these gene product concentrations evolve through time. And here they're flattening out, they're moving towards stable concentrations indicated in this cartoon over here. And that means the parameter sets we chose here caused the pattern drawn here to stabilize. That may or may not be the pattern that the real fruit fly makes. It usually is not. Now, but this is a way we can assess whether a given randomly chosen set of parameter values does or doesn't make the network do the right thing. Now, I have to make a little digression because I want to talk about the shape in parameter space of the values that make that segment polarity network generate the stripes that we see in fruit flies. Here's an old-fashioned Cartesian plot in 3 dimensions showing how 3 radio parameters might vary. The 17th resistor, the 22nd resistor, and the 9th capacitor. And these are values that might be possible for those parameters. And this green dot represents one setting, one set of parameter values that might make this radio work. That's the conventional way to look at such a parameter's value. Here's a different way. We make this wheel plot in which we have 3 axes, one for each parameter, and the inner circle is the lower limit on allowed values, the outer circle is the maximum value, and we just locate the values along these axes and draw, in this case, a triangle connecting the points. And that green triangle is a picture in an alternate fashion of this green dot here. We might try a different set of values out here and find that the radio doesn't work. This dot corresponds to that triangle. Okay, so you can see by the shape of these polygons where the parameter values are in parameter space. And we might do some experiments and find out a variety of close-by choices for these resistors all make the radio work, but this one doesn't. So we would end up with this diagram in this wheel plot of the parameter values that made the radio work, do some specific task. Now that's an awkward way to draw a picture of these dots. Its great virtue is it generalizes to high-dimensional parameter space. Here we go into 8 dimensions, and you can see in this river of sort of similar polygons, pictures of parameter sets that are in the same locale in parameter space, different from the zone where this brown one is. And the point is that we can visualize with these pictures what a parameter set looks like in any dimension parameter space we want. This is what we found for the segment polarity network. We tried out about 250,000 random choices, and we found among them something like 1,100, 1,200 parameter sets that made that network that I showed you, that the experimenters discovered, create the correct patterns in Drosophila embryos. Now, the point is the parameter space is just completely full of these parameters. There's almost no region. There's a tiny region here that is not inhabited by some good set of parameter values. And that's remarkable because we allowed approximately 1,000-fold variation from the least value here to the greatest value there. So not 1,000%, 1,000-fold changes in all of these parameters that govern how this network works. Now here is the really remarkable thing we found, that 1 out of 200 randomly picked parameter sets make that segment polarity network create the right stripe pattern. Okay, so that may not sound like much, but I'll convince you that that is an astonishingly high hit rate. So what does that mean? It doesn't sound like much. You know, if that's how often the IRS didn't audit you, you would be upset. So, but if you think about this in a high-dimensional parameter space, it really changes your interpretation of that number. This red box is one marking off 1,000-fold variation for each parameter. And the green box inside it is one allowing 900-fold variation, 0.9 of the full variation. 0.9 raised to the 48th power turns out to be 1 in 200, the hit rate we found. So 1 in 200 hit rate means that along each axis of 1,000-fold variation, you have a 90% chance to pick a good value for each parameter in turn. And that's an astonishing, robust behavior of this network. That is to say, it almost doesn't matter what the strengths of the connections are, the network seems wired up to make the pattern that it makes in fruit flies. You're supposed to lift with your imagination this picture into 48-dimensional space and imagine this green box as the volume of the good points in parameter space. So at that point, we had only one network that we had analyzed, and we thought we were astonished by this robustness. We thought this might be some kind of fluke. Elie Meyer undertook to make a network of this other famous developmental network involving delta and notched and acute and scute, the proneural genes and suppressor of hair loss and all these genes here. And this network operates to pick out the precursors of a certain set of neurons in fruit flies and in other organisms. And Elie found this network too had about the same 1 in 200 hit rate. This was a common feature then of the two networks that we could study. Remember, it took 3,000 person years to figure out that first network. There's not many networks known today in this kind of detail that you can make such a model, but the two that we found both shared this property that surprised us, surprised me initially. But I now have come to think that this robustness is actually essential in order that a network be functionally heritable. And I want to talk about why that is. So this diagram is a 3D picture of parameter space, and I'm focusing attention on this blob here that might be 10-fold variation across. And this blob is the kind of blob that Elie and I found existed for that delta notch network, within which not only is the hit rate enormous, that is 95% of the points in this blob make the network do the right thing. Not only that, but you can mate randomly individuals located in this blob with each other at random and get, with 95% probability, networks that also work. So this, we think, represents a polymorphic population, and these dots are supposed to be individuals. And this is what we found for that neurogenic network, a parameter space with these kind of blobs all over the place, here and there, located, if you will, like the holes in 50-dimensional Swiss cheese. So, and we're talking about one hole in the Swiss cheese. Now, in this blob, as I said, you can combine the parameters for any points in here at random and get another network that works. And you're supposed to imagine a population like the human population spread out across this blob with all of these individuals having a functioning version of the network, not knowing, some of them, that they're on the frontier of this blob. Now, what that means is that somatic mutations or genetic perturbations caused by cosmic rays or smoking or whatever can move you around and do move your networks around in whatever operating domain they are. And that is without consequence if you're lucky enough to be in the interior. But it can have a heavy consequence, a negative one, if you're at the frontier by kicking you out of the zone of healthy function into another zone where this same network with different parameter choices does something utterly different. It presents as some syndrome at the hospital. And in this other blob, that new action of the network may be done just as robustly as the correct action here. The point is, unlucky individuals can be moved by tiny perturbations from a healthy operating point to a sick one, and there may be, in fact, several or many different neighboring zones that we would call different syndromes that you could move to by somatic mutations. The point here is to think about human health caused by human disease caused by genetic perturbations as arising in countless different ways as you move along the many axes in this high-dimensional parameter space. There's countless ways to get sick. A given network, when it fails in one way, this way could present as one syndrome, and when it fails in another way, it could fail in another syndrome. There's as many ways to get sick as to repair the action of this network by some clinical intervention. I want to mention that the genes I'm talking about in the segment polarity network actually are associated with human health. Foul-ups of the wingless gene often present as breast cancer, and foul-ups of hedgehog are associated with skin cancers. So in order to cope with polymorphisms and make the children's network work regardless of what genes they get from their parents, I think every network must be robust in order to be functionally heritable. And now I want to talk about this new work, probing how could this robustness have arisen. I want to underscore there's a conundrum. If it's true that a network of genes has to be robust in order to get into the natural selection race, you can't attribute the tuning up of the network to make it robust to natural selection. All right, so that's a logical difficulty here. If I really mean this, then this means that the genesis of this robustness has to be something that comes before natural selection starts working on the networks I'm talking about to make them better at some particular task. So this is the task that Edmund Rowe, a colleague at the Center for Cell Dynamics, and I, a founder of the Center of Cell Dynamics, and I, decided we would undertake, and we decided to undertake it in a backward kind of way that I'm about to describe. Bjorn Millard, who looks in this picture as if he was made from plastic, is an actual wonderful UW undergraduate. He was an undergraduate at UW, got his degree in biochemistry, and came to work with us for about a year and did the project I'll now talk to you about. He did almost all the work behind what I'm going to say next. He's now moved on to be a graduate student at MIT. So here's what we did. I wrote a program that spews out an endless supply of biochemically sensible but globally senseless genetic network diagrams. And I designed this first to be incapable of designing a network for any purpose. It just throws random numbers and puts these networks together at random. They do have about the same number of parts as the segment polarity network that we're trying to explain the robustness of. And then, to just cut to the chase, what we did is we made a bunch of these networks, and we analyzed 20 of them carefully, in addition to this one, which is the segment polarity network. This little cartoon shows the segment polarity stripe of engrailed and wingless and patched and so on, that network makes in fruit fly larvae. And then these other networks are among the random ones that my program created, and these are patterns that those networks turn out to be able to make. So here's another group of them, and you can see that they're different in various ways, one from another. They have different numbers of diffusing proteins and different numbers of dimers and different numbers of all kinds of things. And on we go until this slide reminds me to highlight that we only found one network that couldn't stably and robustly make a spatial pattern. So that's a punchline of our work. 19 out of 20 of these networks that we haphazardly concocted at random turned out to be able to make patterns of the complexity that you see, approximately the complexity of the segment polarity network, turned out to be able to make these patterns as robustly, or sometimes much more robustly, than the segment polarity network could do. This is why it's a backward scheme that Ed and I chose to try. We considered specifying some fancy pattern and evolving on a computer networks that could make it. But we decided that would be too hard and it wouldn't tell us anything. Instead, we make random networks, and then Bjorn wrote software to catalog all the patterns that network could make. And then sought among those patterns some patterns that the network could make robustly. And the idea is, or the punchline is, that 19 of 20 of the networks that we tested were as good as the segment polarity network at this kind of a task. One peculiar network that looks no different than the others was incapable of robustly making a pattern. So here's the rest of the 20. And I now want to confess that, I hardly believe this punchline. It is made slightly more reasonable by noting that I actually generated 103 of these networks, and by vote, Ed and Bjorn and I cast out something like 80 of them, not because they couldn't make patterns, but because we could see that they would make patterns for a trivial reason not worth analyzing. So even if they didn't make patterns, 19 out of 100 is still an enormous ratio of random networks that make patterns robustly. And I think that ratio would be something like maybe 85 out of 100 if we hadn't cast out the bad ones. Okay, so I'm now going to put into this talk some details because I want to convince you that we did something. And I'm encouraging those of you who are craving a nap to sleep through the next part. So here's the problem. We put identical copies of these networks in 16 cells. There are then hundreds of different concentrations to keep track of. And we cycle through random assignment of initial conditions, and the combinatorics are simply overwhelming. The number of different patterns that could arise for our software to explore is beyond bound. You can't enumerate the possibilities. But we decided to do this anyway. And so what we did is, with a network fixed, that's going to be fixed through this discussion, and the colors are going to show different gene expression states in different cells. There are 16 cells. And remember, it's the gene expression state that determines whether a cell back in that hornworm is going to be part of the eye or the liver. Well, it doesn't have a liver, but it's, the gene expression state determines what the cell does to build the body of the caterpillar. Okay, I'm going to focus attention on some particular protein, gene B's protein, along this axis from low to high. And for every state that shows up in a stable pattern, I'll just put a mark on this diagram. These 4 blue marks here represent these 4 cells adopting that cell state that has some particular value of this protein B, and the other states have other values of protein B. And now we change the initial conditions, we get a different pattern, we put some more marks here, and we go through this accumulating this diagram that shows how often each of these different concentrations along this axis show up. And after we do this for tens of thousands of initial conditions, we can then enclose this bar graph in a curve like this, and fill it in, and thereby obtain this diagram showing how often concentrations, low concentrations, medium, high concentrations arose in our numerical experiments. So this kind of a diagram is what I expected to see when we did the numerical experiments, that you could get all kinds of different concentrations of a given protein and other proteins with other concentration diagrams, and that we would end up with something like this. That would have made our study hopeless, by the way. But what the picture shows is that if that's what happens for gene B, and the greenness here shows the intensity of protein B expressed in these cells for a whole bunch of different initial conditions, this would show the concentration of some other protein, and we would end up with these numerous possibilities. The break we got, and what made this study work, is that instead of this, what we expected, what we found are diagrams like this. The typical protein was expressed at a very few different levels, in this case just 2. So these are graphs with tens of thousands of initial conditions behind them, and what's important, what deserves these exclamation points, is that there's only a few different expression levels, not all these ones that we expected. So that let us group together the parameter sets, like these 3 across here, that shared the same number of expression levels for each of the gene products. So look at this one for protein F. Each of these 3 parameter sets had 2 distinct levels. The levels were different one from another, but the number of levels was the same. And that's how we grouped parameter sets together. You'll see some histograms coming up in future where each bar, such as this bar, represents a stack of parameter values, parameter sets, every one of which has the same number of expression levels for each of the gene products. So this bar here represents a stack of usually hundreds or thousands of parameter sets that, even though the parameter values are different, the number of expression levels are different. In more mathematical jargon, that means each bar represents an equivalence class, something I'll define in a moment. That is to say, when we group all these parameter sets together that share a number of expression levels and abstract this to this icon represents this whole equivalence class of parameter sets. And we thereby partition all of this huge parameter space into a manageable number, it turns out a manageable number, of equivalence classes of parameter sets. Each equivalence class has the same, shares these numbers of expression levels. Let me take a moment to say what an equivalence class is. It's something I used to teach students in mathematics, and it's finally come in very handy. Think about fractions in 3rd grade that you learned about, 2/3 and 6/9. These are all different names for exactly the same thing. We think these are equivalent fractions, and that's because 3 times the numerator is the same as 2 times the denominator. And all these fractions strung out here infinitely far for all the different names for 2/3 constitute a rational number. One rational number is that set of equivalent names of one rational number. We say mathematically that these 2 rational numbers these 2 fractions are equivalent if this formula holds. And in a similar way, we say 2 sets of parameter values are equivalent if they're associated with the same network, of course. And if in addition, for each gene product that the network makes, the same number of stable expression levels arises across all initial conditions that we try when trying tens of thousands of them. So that trick, so I haven't actually told you the real mathematical details, but I've suggested them, that trick of partitioning a too vast collection set into a manageable collection of equivalence classes turns out to be, believe it or not, one of the most powerful tools mathematicians have for constructing complicated, qualitatively new things from previous simple things. They made the rational numbers from integers using this construction. They then made the real numbers from rational numbers using exactly the same construction. And to give a slightly fancier example, they made the tangent space attached to each point of a differentiable manifold by defining an equivalence relation among all the curves that intersect that point, and thereby made the mathematical substrate or structure upon which Einstein hung general relativity. So this construction technique has a long history, and it's very powerful. We used it to partition a gene network's huge parameter space into a collection of equivalence classes of parameter sets, and that let us do what I'll show you now. Let us catalog the patterns that a network can make. So still the network is fixed, and I'm amplifying a little bit what one of these histogram bars means. It stands for an equivalence class. Here's the icon or signature of that equivalence class, and the purple line meandering here represents one gene expression state with high, high, medium, high, high, low levels of that gene, and another state, color-coded yellow, would have different levels, still in the same equivalence class. And what we did with our software is find all the 6-cell surround patterns that arose in this network that it stabilized when we cycled through countless initial conditions. And once we had these patterns, we could figure out which striped patterns, the kinds we're after, could be made by tiling with these 6-cell surround patterns. And it let us forecast the striped patterns that could be made using all the parameter sets in such an equivalence class, and that forecast turned out to work well. So in the pictures coming up, you're supposed to imagine each little bar in a histogram as a surrogate for a great menagerie of patterns that equivalence class can cause this network to make. If you took the nap I suggested, it's time to wake up. And I want to remark that this is a big calculation. We only looked at 20 networks because we ran out of computer time. The diagrams coming up summarize a big calculation, about 3 months on a cluster of 300 really fast Linux processors. That's about 75 CPU years. That means, so this laptop is about 3 times slower than any one of these processors. That means if I started this calculation on this laptop when George Washington conquered New Jersey and then went on to found this university, the calculation would still be running. Okay, it's a long calculation. And so here is a fully filled out picture of the patterns that were made by members of this equivalence class, and a different bar in the histogram corresponds to a different equivalence class, different set of patterns. In the diagrams coming up, there will be a picture, a color-coded picture of a pattern, one among many that the network made, and this is the hit rate. Look at that hit rate. 1 out of 6 choices, random choices of parameter values, cause this network to make that pattern. It's in some mysterious way, the ability to stabilize this spatial pattern is wired in the topology of this randomly generated network. By the way, in the diagrams coming up, the blue bar represents all the parameter sets that gave rise to boring patterns, to patterns in which every cell had the same expression state. So it's a pattern, but it's a null pattern and uninteresting. All these blue bars are the same heights in the pictures coming up. We normalized to them. So here's a parade that I'm going to zoom through of 20 networks. That's the segment polarity network and the pattern that it makes, the one that's appropriate for fruit fly development. And here is a number of other networks, and it takes a long time to stare at these and puzzle out what may be going on with them. Maybe the only interesting one is this abject failure. 1 among the 20 didn't make any pattern robustly, any stable pattern robustly. I do not know why, but I guess the hard question here is how could you design a network at random incapable of forming complex spatial patterns robustly? So this, I think, is interesting, that with no optimization, randomly designed senseless gene networks are capable of forming spatial patterns that are as complicated as and as robustly as the highly evolved segment polarity network makes its patterns. And that, to me, is quite unexpected, because when we first came across this robustness, I spent a great deal of time trying to figure out how would you make a network with that feature. Our human-made technology like this doesn't have that feature. If you change the parameter values here, the radio won't work. How would you design a network with this remarkable feature? It turns out you don't need to do anything at all, apparently. Now, so let me amplify this a little bit. If, you know, for the technology that you come across in Walmart, it is usually correct to assume that somebody thought it up, designed it, and got it manufactured in China. That's a correct inference, probably. It is not correct, however, and this is a hard thing, I think, to wrap your mind around, it is not correct to extend that inference to the most astonishing technology you're likely to come across at Costco, such as, for example, as an onion. You know, the biological mechanism has astronomically greater sophistication than any radio circuit like this. That does not imply it was created by something fancier than the designers for Walmart. Now, this pattern, this network is robust. It makes a complicated pattern. It's as complicated as that radio. And its robustness begged the question, what designs that robustness? What creates it? And the punchline is, this apparently amazing design feat turns out to require no design at all. I want to make, that was a quip, I want to make a serious suggestion here. And that is that an organism with thousands of genes in it, some of them transcription factors, many of them interactive with other genes, will have accidentally countless inconsequential gene networks that arise in it that make all kinds of patterns in an organism. You probably are full of expression patterns of genes where those patterns don't matter at all. So it seems to me that by accident, such patterns can easily arise, and in fact, it would be almost impossible to suppress such patterns in organisms having thousands of genes. Those patterns are likely to be made already robustly as a consequence of what I've just told you, that robustly concocting, that accidentally putting networks together yields networks that makes patterns robustly. And those robust patterns that are initially inconsequential are available for natural selection to hang downstream consequences on and cause those patterns, like these patterns of the engrailed gene in this fixed fruit fly embryo, to cause the expression of downstream genes that make bristles grow out of here or make something happen here that has a phenotypic consequences. So in other words, if your future descendants absolutely require stripes, it's likely that they already have accidental patterns of stripes expressed robustly that they could hang pigmentation stripes on. All right, so this work came from the Center for Cell Dynamics that I'm the director of, but all I do is participate with these geniuses here that work at this center. This, by the way, is a ctenophore, one of the marine invertebrates that flourishes off the dock at Friday Harbor Laboratories. What we mostly do in this lab is do concrete, real experiments to observe what happens in these interesting developing embryos and try to puzzle out what particular real gene networks actually do. The work I've talked to you about tonight has been highly abstract, but I hope nevertheless interesting. So thank you very much for your attention, and that's what I have to say.